Okay, let's do B cell receptor signaling. Not as complex as some of the other signaling that we have just looked at for the T cell receptor, but still complex enough. All right. So first you got your parts of the B cell receptor, and so that'll be the two heavy chains and two light chains of the B cell receptor, which differs from an antibody only because it has transmembrane regions on it. And this would normally be an IgM type antibody, but the surface mounted kind, that's what a B cell receptor typically would be. It's going to associate with Ig alpha and beta right over here, each of which has an ITAM on the bottom. All right, and the ITAM is going to associate with one of these Sarc family kinases. This is three examples. You don't need to give the example. You can just say a Sarc family kinase, and that'll be fine. The Sarc family kinase is going to associate with and phosphorylate the Ig alpha and Ig beta, basically. Once we have that, then we can bind to psych. And so psych is going to be similar to ZAP70. It's going to only be activated when it binds to a phosphorylated ITAM. And psych is going to be a kinase, and the psych kinase is then going to go off and phosphorylate other things. Now, over here, let's see. The B cell is going to have a co-stimulatory receptor, which is fine. And some of these possibilities are going to be CD19 and CD21. Those are good co-stimulatory molecules that form a complex. And let's see, down here our Sarc family kinase is actually going to go over and phosphorylate CD19, and once it is phosphorylated, it will then recruit a PI3 kinase, which will turn PIP2 into PIP3, and once you got PIP3, then you can do all that same other stuff. And yeah, with the PIP3, we can activate PLC gamma, we can do the VAV thing, and so I think I'll actually just chop this block out since we've covered the T cell equivalent of this, and this is only slightly different, but I don't want you to get confused by it, so we'll just leave this last bit out. And then the last thing that we need to know is that this is how B cells detect when the battle is over, essentially, when they've got their B cell receptor and it is attached to some kind of antigen that's attached to an antibody that's attached to another antigen that's attached to another antibody that is attached to Fc gamma receptor 2 type B1. Fc gamma R2, that's Roman numeral 2 like II, B1. That has an ITIM on the bottom of it. That's why it's a little red rectangle instead of a yellow rectangle. Still a place with tyrosines that can be phosphorylated, but it's going to lead to inhibition. And so SHIP is the thing that's going to bind to that. The Sarc family tyrosine kinase will come over and phosphorylate this if it is nearby. So only if the Fc gamma receptor 2B1 ends up in the same vicinity as the activated Sarc tyrosine kinase, then it's going to get phosphorylated. So only when there's a collection of antigens and antibodies. Anyway, SHIP's going to bind to that, and then SHIP is going to take PIP3 and it's going to turn it into PIP2. So it's going to dephosphorylate the PIP. Yeah, take off one of those phosphates. And now, since we have PIP2 instead of PIP3, we no longer have anything to bind PLC gamma or VAV or BTK to the membrane. They just end up floating around and not doing anything. And these things are not active unless they are bound to the membrane in these two cases and activated by a phosphate and bound to a membrane in the case of PLC gamma. All right, so that is how we turn on and turn off the B cell receptor signaling, essentially. And I hope this helps.